Hello, everyone. Today, I'm going to present. Basically, do it's not going to strictly be my research. Basically, it's not going to be the results of my research. But today, I wanted to share with all of you basically what is the field in which I'm doing research, and I really want to give a perspective of where I think this field is going, what has been done already in the past, and in the, the I would say the past decade and where basically my research fits in, in this domain. So I tried to make the presentation as digestible as possible. So I tried to reduce jargon, and I tried to make it more like basically like a review of the state of the art rather than presenting actual results. So again, it'll be probably like a bird's eye view kind of thing about the field. And obviously, I want to have this as interactive as possible. So if you have any questions, feel free to put them in the chat. I'll take a look at the chat while I'm presenting. So let me share the screen. So hopefully you should be able to see the screen. Okay, so the agenda for today will be this four topics. So I will start. I'll kick it off by basically describing what is a biosensor, what makes it different from a general sensor. I will then make a panoramic of what is the field of Salivaomics, why is that relevant, and what is saliva differs from other biofluids that we could be interested in in the field of diagnostics. Then I will introduce the term lab in the mouth, obviously, detailing the different aspects of how, this lab in the mouth is helping with the new, the new, basically the new advances in diagnostics. And lastly, I will, basically show you what I think could be really the future for this field. And, maybe, potentially, what will be the challenges associated with that, and what will be, the big unlocks, the big benefits that could, potentially bring. But first, little introduction about me, just a few minutes. I started off, with my bachelor in my hometown. I started with, studying chemical and biochemical engineering because I was really interested in chemistry. And then I decided to switch to material science um, because I was particularly interested about how, the materials could be, new innovative materials could be used in the field of medicine. And so that really, that's basically what pushed me to do the master at Georgia Tech. Uh, but I actually ended up not working in the medicine field, but more in the robotics, where I was developing stretchable and flexible sensors for robotic systems. And then I got a possibility to basically, fulfill my dream, my academic dream of working at the intersection between engineering and medicine during my PhD, where I am now currently developing a point of care device. I will get into that later for oral health assessment. So basically, a device that could tell you, how good or bad it is, is the status of your oral cavity. Um, and so, yeah, my research interests are really at an intersection between flexible, searchable electronics and biochemical sensor you know, with a good focus also like on wearable devices, because I'm really interested in wearable, even if, I'm developing a point of care device now. So let's start with the basic question that, what you should, what I would like you to know, if you don't know it already at the end of this talk. And it's basically, what is a biosensor? So a biosensor by definition is a device that is able to translate a biological reaction into a measurable signal that we as humans, or I'll say machines, could interpret and use for specific purposes. And so there's a lot of biosensors in our life, and I'm pretty sure that some of you have interacted with those. I mean, at least during COVID, we've seen a few examples, but I would say the most widespread example of a biosensor is the glucometer. So the glucometer basically works. The biosensor is in that strip that you see plugged in into the device. So the strip is actually the biosensor. The whole device is more like a biosensing system where you have a readout device that is the little device, and then you have the biosensor that is the strip. And so what it does, it basically, takes the blood from, takes a sample of blood, and it can tell you, your glycemic concentration, using the biosensor that is plugged in. But this could sound complicated if you don't know exactly how that is made. So let's try to, take apart the different elements of a biosensor. A biosensor is basically composed of 4 different elements. The first one is the analyte. The analyte is basically what we want, what we're researching. What is, the molecule that we're trying to find in a specific sample. Could be a biofluid, could be a wastewater, could be even like a solid sample. There's some biosensor for solid samples. And so the analyte tells us the type of molecule that we're researching. If we're looking at a DNA string, is very different than if we're looking at a glucose molecule, for example, or an ion, for example, if we're looking at iron or cobalt and so on. And so the analyte is really the first thing that you want to know, you want to be sure about when you're developing a biosensor, because everything else really depends on your analyte. And in order for us to detect, to basically see that biological reaction happening, we need to have something else to detect the analyte, and that is a receptor. Receptors can have a lot of different shapes and natures. And so, the most used ones are enzymes. So for example, the glucometer that you saw before, that uses an enzyme as a receptor, that is glucose oxidase. That is basically what allows us to see that, to actually make that biological reaction happen. But there's also antibodies, for example, the one that we used for detecting COVID, for detecting the COVID spike protein in during COVID times, and then you can use also nucleic acids and cells. And so now you have an analyte and you have a receptor. So these two binds and allow you to see that biological reaction. But how can you actually see it? Well, you need to have something called a transducer that basically does the translation part. So take the biological information and put it into something that we can, analyze and see. And so transducers there's several types. I would say that the most used ones, the ones that are more widespread, are electrochemical sensors. So electrochemical transducers allow you to translate a biological reaction that could be a redox, so like a phoretic or non-phoretic reaction, into a measurable signal. Optical transducers do something similar, but in this case, you're not measuring, you're not getting as an output. You're basically monitoring the optical properties of your system. And same applies with thermal and piezoelectric. In piezoelectric sensors, for example, you're measuring a change of mass. So for example, let's say they have a receptor, and that receptor is on a little, miniature scale. When that receptor binds with the analyte, what you see is a change in mass. And that is how piezoelectric transducers work. And so what you see below is basically an example of that. What I'm saying, we have glucose on the left as the analyte. We have that strip that is the whole transducer, because that strip has a transistor in there that is basically our transducer. And the resistor, on top of the resistor, we have the receptor. And the receptor is that layer that you see on top of the orange elements. And that receptor is the one that binds with the analyte. The transducer translate that, and what we get out of it is the signal output. So we ultimately want to receive something that we can interpret, understand, ideally on our desktop device or on a mobile device, so we can store the data and use the data for later applications. And so this is really, in a few words, what is a biosensor? What you should really remember about a biosensor, and also what makes it different from a sensor. The sensor is different from the biosensor because it doesn't have the bioreceptor elements that, that basically makes the biological reaction happen. So sensors do not have any biological element in them. And so that's why they differ from a biosensor. Um, so there's a lot of biosensors that we have seen in our life, and they've changed in a way that it's probably very simple to what we've seen already in the past. And that is computing. So if you think about computing, we have started with something like the device on the left. So this, big machines that could, take up the space of a room. And then we reduce the form factor of those machines into portable devices like our laptops that we can, bring to work and bring back home. And then ultimately, we're now more and more going into the wearable format where we have this machines, these computers that can fit inside a clock. And so now we have, this smartwatches have a lot of computing power that can, do a lot of amazing things that were not even remotely possible even 20 years ago. And so if you look at this reduction in form size, you will notice that something similar is happening in the biomedical devices space. So we've started with something benchtop like this, like the glucometer that you put on the desk, and then we've reduced the form factor to bring it basically into a wearable format, where now you have a portable glucometer that you can carry around and you can use whenever you need it. And now we're starting to see with Abbott, this is a device from Abbott. Now we're starting to see the first appearances of wearable devices. So where you don't even need to bring the device anymore, it's more like the device that is coming with you um, on your arm, like in the case of the Abbott sensor, where you have a needle that is inside your skin and continuously gets the blood from your arm and allows you to take the analysis that you normally do with a portable device. And so again, what we're seeing here is something that we have witnessed already, and we are currently witnessing with computing. And so going from benchtop to wearable formats. Biosensors are, as I said, ubiquitous. There's a lot of different sensors that we have that we're currently interacting with in our life. And so this is a figure from a paper. This is a great review paper that came out 3-4 years ago about wearable sensors that can show you in a really easy way, like, really visible way, how we could really use sensors everywhere in our body. We have sensors that can be used in the mouth as, mouth guards, and I'll be talking about that today. But we have sensors that can be embedded even in smart in contact lenses. And so obviously, the most the biggest advances in the field have been made in the field of sweat sensing, because it's really easy to access. And so we've seen a lot of different examples of sweat patches and sweat sensing devices that you can attach in your skin. But we've also seen examples of sensors that can be used in textile formats. So now you are embroidering some sensing fibers in your textile elements, and you can wear those, and you wouldn't even feel the difference. But now you have something that is embedded maybe in your shoe, on your socks, and now it can sense stuff from your skin. So again, the field is moving really quickly, and there's a possibility to gather information from our body pretty much everywhere. And research is showing this already. But it would be too long for me to tackle all of these elements at the same time. And so today I want to restrict a bit the field of wearable sensing, wearable and point of care sensing, to a specific part of our body that is basically the oral cavity. And so what I want to do today is introducing the field of salivomics. Maybe some of you are familiar with the term already. maybe some are not. Salivaomics as a term basically was born, I would say, not more than 20 years ago. So it's a rather new field, but we've seen a lot of research coming out in this domain, especially after COVID, after basically people started to use the saliva-based tests instead of the swab test for to detect COVID-19. And so, what is salivomics? Salivomics is really using saliva instead of other biofluids. So, for example, instead of using blood, that is the gold standard for the majority of tests. So instead of using blood as a biofluid, we use saliva. Why would you use saliva instead of blood? Well, first of all, saliva is easily accessible, so it's not like blood is not painful to obtain. It is continuously renovated. So if you think about it, you produce a lot of saliva during the day, and so if you want to take an average of what is the concentration of a specific molecule, you can do that because the saliva is continuously renovated in your body. So as I said, it's non-invasive, it's easy, stress-free, it is repeatable, and also it doesn't carry any risk of contamination, as it could be, for example, the case for blood. The only thing you need to be um, aware of with saliva is that it is really dependent on what you eat. So maybe if you want to take a test, unless your desire is to measure something that is related to your diet, you maybe don't want to take the test right after you've had lunch or any type of meal. But apart from that, I would say saliva is an extremely promising fluid when compared to others that we have already. And saliva has a lot of different molecules in it, so you can really gather information from several different subdomains of you could be interested to. So, for example, we've seen research coming out where you could detect to the single molecule. So we're talking about attomolar concentrations of stuff in saliva. In the same way as obviously, with different devices, you can detect much bigger molecules, like, for example, glucose and cholesterol. So really, saliva has a lot of different molecules that are interesting, and they could each be targeted in a different, in a specific way by using the right receptors, as I've shown before. Really, the key is about finding the receptor to target a specific analyte in your saliva. So as I said, the field of salivomics, salivary diagnostics, how you want to call it, was really, born, I would say 20 years ago, when, first examples coming out with some biosensor that you can see on the left. Now, in this image, you can see it's basically like a time scale where you can see how we've evolved. So we've started, I would say the first example coming out in a paper on Nature is the one you see in image B. That is a tooth enamel biosensor. So this biosensor is basically a strip, is a resonant antenna. So basically, it's a device that you can interrogate in similarly to what you do with your phone. You can send, you can send some waves to the sensor, and the sensor sends the waves back. And depending on, there's a receptor on top of the antenna. That receptor binds to a specific molecule, and so depending on what is the concentration of your analyte, the response that you get back in terms of waves differs. And so you can draw basically you can basically draw a curve that can tell you what is the concentration of a specific analyte, depending on the response of the sensor. So that was really the first example of, okay, we can create a sensor that we can put on top of a tooth and people could potentially wear it. Now, obviously, wearing a sensor on top of your tooth is probably not the most practical way to do, diagnostics. And so that's why we have evolved from that initial idea into something more practical. So now we have, for example, mouth guards that could have, that could house sensors. And that's way more practical because you can remove the mouth guard, you can wash the mouth guard if you need to, but you're still going to have the sensor there and it's not attached to your tooth. And so we've seen examples different mouth guards that you can wear, and that can give you information about different different analytes. But really, we've also seen a lot of new research coming out relatively to point of care devices. So for now, I've only talked about wearable devices, but in some cases, you don't really need a wearable format unless you're tracking something that consistently varies during the day. And so it's much easier to just carry out a test in the same way as you would do in a laboratory. But in this case, you could do it comfortably at your home without having to go to the hospital because you have this point of care device that you can use and uses saliva. So you don't even have to finger prick or, take blood out of. That is normally something that you would do in a laboratory setting. Um, so yeah, this is again is to show you how the field have really started to put out research, a bit more than 10 years ago. And we have already advanced a lot because a lot of, research is pointing in this direction for either point of care or wearable devices. So let's now focus on only the wearable part of this because probably the one is most exciting about this field of research. Not because the point of care is not, but because of the point of care device. Some of these devices have already entered the market. And so there is a maturity of the field that is basically acknowledged. And so there's many more players looking at that than we have on wearable intraoral devices. And so today, as I said, I wanted to introduce the concept of lab in a mouth. You've probably heard already about the concept lab in a skin on a skin. If you have dealt with sensing, wearable sensing, and it's basically the concept of having translating all of the techniques, all of the capabilities of a laboratory from a laboratory setting to a wearable format. So Loveless can basically takes in, microfluidics to get the sweat and drive it to the sensing element and then carry out the sensing and send out the response, also send out the data to a mobile device where the data can be interpreted. So you have all of the functions already in your body. So now we are doing the same for saliva. And so we're calling it lab in a mouth. What is that really about? The concept of lab in a mouth is using the oral cavity as the house for sensors. And there's a lot of different sensors that can be created and that can work in an oral cavity setting. So, as you can see here from the image, we have a variety of sensor for a variety of different targets. So we can detect viruses in the same way as we can detect bacteria, drugs, electrolytes, and metabolites when we're talking about the biochemical sensors. But then we can also, and that's the first one that I'm going to focus on, just because it's the easier version of the sensors, because are those that are not biochemical in nature. And so we're talking about clenching and movement sensors, and also temperature sensors. But let's dig into these two different sensors. So these are sensors, first of all. These are not biosensors, but I still wanted to make an introduction on these two because I think it's important to mention these devices. So intraoral physical sensors, what they do, they monitor the change of a specific physical property. So if you know what bruxism is, if you suffer from bruxism, you know how difficult it is to detect this pathology. And so what people came up with some research groups has came up with is basically creating a comfortable substrate that you can wear. So like a clear liner or like a mouth guard that you can wear. And that has force sensors embedded in the device. So now what you can do is you can go to bed, you can wear these devices, and these devices are able through these force sensors that are literally some printed foils. Okay, you can embed the sensors on the substrate, and now you can wear a device at home, and the device can measure whenever you have the two part the teeth touch each other, and so you can monitor that throughout the night, and you could potentially using some algorithm detect whenever you had some bruxism events. And so this is really powerful. Is a really powerful way to detect a specific pathology without having to go, for example, to the clinic and stay there one night to be examined, or even buy expensive machinery to get home to be diagnosed this pathology. So this is a really great example on how we could potentially use physical sensors. Another interesting use case that you see here in figure D is using these devices for all of those sports. And I'm thinking about, American football, where you can get concussions. Because in the mouth, you can measure, all of this, shocks by embedding a gyroscope that can tell you, the acceleration and deceleration of your mouth in a 3D space. And so you could easily use this to monitor specific events and potentially issue some warnings whenever, an athlete has got has got hit by something. And so that could have potentially created a concussion event. And so that's really important. That's a device that um, could be really useful to avoid some of, some really bad consequences out of, sports. And you can see here, sometimes, a question that I get asked a lot is, how can you fit, how can you fit all of these functions on a clear aligner? If you know what a clear aligner is a really, little piece of plastic that you wear. And so how can you fit all that into, all the sensors into that element? Well, microelectronics has really come, very far into, like, in terms of like the form factor. So now we can have this tiny chips, as you can see in the slide, that can, have a lot of different, capabilities, and can allow a lot of different, detections. And, same applies for batteries and other electronic elements that you need on, to perform sensing and biosensing. So that's why we can do this now. And we weren't able to do it, 20 years ago, in the same way as we've done with computers. So um, the second section that I want to look at is that of electrolytes and metabolites. Um, the reason why we are differentiating here, um, it basically has a specific meaning. So there's some molecules that are easier to detect when compared to others. So if you're looking at a specific, mRNA, or if you're looking at glucose, the concentration, probably the concentration of these two analytes will be different. And so depending on the concentration of the analyte, the accuracy of the receptor they will have to use will also be different. And so that's why we are characterizing this based on the analyte. As I told you at the beginning, the analyte is really what makes the difference, what you really need to measure. Okay. And so we're looking at electrolytes and metabolites because those are the easier ones. Let me say easier ones to detect. And that's what usually people start from when developing sensors, unless they have already, the capability in the end, the know-how to get into other analytes. And so a few examples of these are of these sensors for biochemical analytes. The first one that you see, panel, basically all of these panels, the concept is pretty much the same. You're using either a clear aligner or another device that you put up in the poly, or in the case of figure H and figure E, um, you're using a pacifier. So in this case, you're basically functionalizing something that already exists by adding chemical sensors to it. Really clever move. And so all of these are measuring specific electrolytes. So for example, in panel A, we're looking at lactate. In panel B, we're looking at uric acid. In panel C, for example, and in panel C, we're looking at glucose, and sodium in panel G, and so on. So, as you can see, the difference is really on like how you wear the device. Um, but the end goal is basically the same. The idea would be to measure for a specific amount of time the concentration of a specific analyte. So in this case, if we're interested in knowing what is the concentration of sodium in a person's saliva, what we want to do is create a device that is able to tell the different flavors, basically, to discriminate between different foods by just using the sensor. And so that proof of concept has been shown in the figure of the panel, the figure G, where basically the authors were able to discern, based on the pattern of the concentration of sodium, and they were able to tell the difference between a potato chip, a chicken soup, or a bag of juice. And, similarly, you can see in panel C and D, you can see the difference in glucose whenever, glucose was present in specific concentrations. And so these devices are able to work for I would say a prolonged period of time because it's not a punctual analysis. It's more like a time-based analysis. And so these devices were able to work for, up to 30 minutes and constantly measuring the concentration of a specific analyte and tell the difference between, healthy people and patients that had, some specific pathologies. As you can see, for example, in panel B, where just by looking at the graph, you can tell what an healthy person and a patient with hyperuricemia had in terms of like concentration of uric acid. The the second a second a second section is devoted to drugs because saliva can also be used to do drug detection. And so again, research groups have started to come up with really clever solutions to monitor the concentration of drugs. And so let's dig into that and take a look at what people have done. So the first one is basically a detection that is colorimetric. And so this is not, for example, a wearable sensor. This is a point of care sensor, but it works in a very simple way. There's a molecule that changes. There's an element that changes color. In this case, it's iron. And in the chromophore that is MDCH. And so you insert a sample, and that changes color, and it can tell this based on a colorimetric detection can tell you the presence of a specific analyte and its concentration. The figure in the panel, figure B, is an iteration of that original idea of embedding sensors on a tube. This one is a tiny sensor that is based on two rings, and it works in a similar way as I said before. You basically can send wave to this. This is antenna, so it's a resonator. You can send wave to this resonator, and what you get back is a response that depends on the thickness of that layer that is between the two antennas. Now, that layer is sensitive to the concentration of alcohol. And so basically that will change its thickness depending on the concentration of alcohol. And again, you would be able to distinguish between different drinks based just on the measure of that, of those waves that you get back. And be able to discriminate between different drinks. Another interesting idea, and I say interesting, it's because it's not a wearable intraoral sensor per se. It's still a wearable sensor, but it's one that you can wear as a ring. And I'm talking about figure D. In this case, the authors came up with a clever design of creating a ring that you could basically open. It had a electrochemical sensor inside a ring, and you could deposit saliva on the sensor, and it could, instantly tell you, we're talking about, the time of 30 to 60 seconds, can instantly tell you, the concentration of alcohol in your blood, and basically, can discriminate if you're drunk or not. And same with, THC. So if you have assumed, marijuana or not. And so that could be used, for example, um, to do some personal screening whenever you're going out and you want to check, concentration. Maybe you don't have anything to measure and the concentration of alcohol at hand, but you could have a device like this that could, easily tell you if you're suited or not, if you're, within the limits of consumption or not. And so again, this is a proof of concept, but that's, the idea of what we could potentially, see in the future. Another example in this case of a point of care device is the one in figure F, where the authors have created a point of care device. So it's like a miniaturized by a sensing system that could tell by depositing a drop of saliva on those strips that are disposable. So you can keep the device, but you can change the strips whenever those are used. Can tell you the concentration of methamphetamine with a single drop of saliva. So different approaches with the same end goal of doing drug detection through saliva. The third and last one that I want to look at with a specific example is that of viruses. And so we all know how, painful it was, not really painful, but well, definitely not comfortable to do, this, to do the swab test, to test for COVID during COVID times. And so why couldn't we use saliva? Well, people thought about it, and they actually developed sensors to do so, with, in some cases, a pretty good accuracy as well. I would say at least comparable to the swab tests. And so in this case, the authors have basically repurposed an already existing glucometer, uh, and by basically telling the glucometer, by basically functionalizing the sensor in the glucometer, they were able to tell the concentration of COVID-19 using a specific reaction between the some aptamers, um, some magnetic nanoparticles, and the COVID-19 needle spike protein. And so by basically using the some amplification steps and a magnetic separation within a test tube, they were able to basically add sucrose to the formulation and use invertase enzyme to turn sucrose into glucose and basically measure the concentration of glucose in a glucose strip. And so by not even creating a new device, we were able to repurpose something that was already existing to measure basically COVID-19 in saliva using an off-the-shelf glucometer. Really amazing job, especially considering that the authors here really didn't create any new device. It was all about creating doing the surface chemistry to allow the specific biological reactions that we're looking for to happen on an already existing device that had a glucose oxidase layer on top, as is always the case with those glucometers. So that was really the last example of what can be done in the field of saliva detection. Analyte detection in saliva and salivary diagnostics. Now, I want to end talk by talking about what could this field be in the future. Okay, so I showed you today. I started by telling you what is a biosensor, how sensors and biosensors have changed, in the past, from the past to today, and how they're probably going to change in the future. Um, I've I've talked about what is Salivaomics, why is Salivaomics interesting, why is Salivaomics promising as compared to other biofluids, and then I did a little review of what people have used saliva already in research to basically detect specific molecules and create wearable or point of care devices to perform these diagnostic tests. So now let's look at what this could be in the future. So if you think about the different elements to build the smart health system of the future, the elements are pretty much already there. It's all about putting all of these elements together to make them work in a seamless way that could bring a lot of benefits and a lot of interesting and promising avenues for both patients and clinicians in the future. So let's start from a very simple element that is the sensor itself. So we've seen that we can now embed sensors in mouth guards, in clear liners, on rings that we can wear. Okay, so everything regarding the substrate and the sensor integration has already been proven in research to work. Now, we also have a lot of different transducers. So we have the possibility to try to detect a specific molecule with different mechanisms. Okay. And so maybe there are some cases in which optical detection is not possible. Is not possible to use optical detection. For example, saliva is a really turbid fluid, is not clear most of the time. And so sometimes it's difficult to do optical detection in saliva due to the color of saliva itself. And so maybe there's something else that we should use, like an electrochemical sensor. But we have already developed, individually these sensors that work for specific use cases. And so now we have the sensing element, we have the substrates where we could put the elements. We have saliva that is always available. And now we need something to read basically the reaction. And so there's a signal and data processing that you see now here in the figure that is getting cheaper and is getting smaller day by day due to all the advances in electronics and microelectronics. And so now power sources are getting smaller, are getting easier to integrate. So in some cases, there are some sources that could be found inside our own body. So there's some examples of some devices that are able to operate without batteries. And so removing the risk from batteries because we're using, power harvesting from maybe our body, from the temperature of our body, from actually even using some biofuel cells, being able to use specific reactions to produce the power needed to power a specific circuit. And so again, power sources are also rapidly evolving. Batteries are rapidly evolving. They have different form factors, different sizes, and they can be easily integrated with microelectronic circuits. And so this brings all of the hardware together. Now we have this hardware. It's small. It's integrated. It can be put in the body. Can be worn during the day. Now, what do we need? What else do we need? We need an infrastructure where we can get all the data from the sensors in a continuous way, and using algorithm to process this data continuously to basically give informed to be able to create a database of information that clinicians can use to take action and to give informed decisions and for the patients. And so now, if you think about this, these elements that are always interconnected, that are using some AI tools that can, extract the most relevant information. And all of this can be done in a seamless way and actually even in fashion, if you think about it, because now every patient could have their own device that they could, wear at home and that can provide information for them or for the clinician. They can decide to share this information if they want to. So, this is how, the field, this is not me, this is actually a figure from the review paper where I've taken, most of the images. This is where the field is probably going. There's some elements that are already there. I would say that, most of these elements are already there. What is really missing now is the links, the link between the different elements. And this is also, and I'll be closing the talk on this note. This is also what I'm trying to do with my PhD research. So during my research, I said it at the beginning. So I also want to reemphasize it at the end. I am developing a point of care device. And so it's a device that you can use in the setting, wherever you want to deploy it. It's basically a device that is as big as a smartphone. And you can use it to assess the status of your oral cavity. So now my idea, the idea behind my PhD is basically, developing everything from the ground up. So I'm developing the sensor. I am developing, I'm basically using some specific materials to detect specific analytes. And so I'm putting that together. I'm putting together the hardware. And the idea would be basically to get, data that can be sent to a database where, I could potentially apply some AI tools and extract some relevant information. And so in the little environment that is my research setting, I am already trying to apply this concept and trying to go, toward what I see as the future of health. And now, the future of health, in my opinion, is something that is continuously interconnected, that uses devices that we can wear, and that it is always providing, a feed of data that we can decide to share or to, keep for ourselves. But if you want, we can share it with clinicians, and then we can, apply a lot of different interesting tools on big data, where we could get really good insights into our health. Because now, maybe our data are being compared with some other data, and we can get some really interesting information. So in my opinion, and I'll be closing here, in my opinion, sensors are a really important element, a really important building block of the health system of the future. Um, because all of this, amazing software tools that we're developing are only useful if you're getting the right hardware tools that allow you basically to gather, to harvest the data that you need to use to take the decisions. So I'll be closing here. I'll look if there's any question in the chat. And yeah, that was my presentation for today. Hope you liked it.
That was awesome, Ricardo. Thank you. I actually had a quick question if nobody had one right now. Um, so I guess my question is, what are these, like, obviously for different purposes, and if you're in different regions, like, or like an oral sensor versus like a textile sensor versus a sensor elsewhere, probably are comprised of different like biomaterials and things like that. Is there like a kind of generic biomaterial that people use when they're developing these sensors, and like specifically for the oral ones? Like, do people test to see if there's like an immunogenic response that's coming from the presence of those sensors? And like, yeah, I guess, well, yeah, pretty much that.
Yeah. So that's interesting question. Basically, there's an easy answer. Yeah, there's an easy answer that is most of the time, research groups start by using enzymes and antibodies. They're battle tested. They've been used for a lot of different applications, and in most cases, they can be used also for multi-sensing platforms. So you can have, some platforms where you can embed, different biomaterials and get simultaneous detection of different analytes. So you don't even have to change the sensor. You can just develop, a multi-sensing platform where you use the different biomaterials. It really depends on the availability of these biomaterials. Most of the time they are available, but the choice is also driven by the cost and the reliability of these biomaterials. So for example, something that we've seen is that um, antibodies and I would say, yeah, antibodies and enzymes, they tend to have some batch to batch variability. And so now when you need to develop a sensor, you need to develop something, unless it's just for research purposes, you need to develop something that is reliable and can be reproduced and can be replicated across a million sensors. So you need to have something that doesn't have a lot of variability. And so now what some research groups are looking into is how can we, you know, create something that is basically, better in terms of like cost and variability when compared to, for example, enzymes and antibodies. So there's a really cool niche field that I'm using some of these materials. They're not my materials, but are basically biomimicking materials. So for example, there's some materials that are called nanozymes. There are some metallic nanoparticles that mimic the behavior of some enzymes. And so now you have something that is not subject to denaturation, that is not as expensive as an enzyme, and could potentially have even better variability. And so same applies for the antibodies. There are some research groups that are researching the so-called plastic antibodies. They're called molecularly imprinted polymers. And so they're basically polymers that are created with the target molecule. When you're creating the polymer, the target molecule is inside the matrix of the polymer. And then when it gets created, when the polymer gets formed, the analyte is removed. And so what you get is a cavity, and that is, shape defined. And that is basically, the aim of that is to mimic the behavior of antibodies. So sorry, long reply. The decision on the biomaterials depend on your target analyte, depending on the cost, and depend on the final application of your device. So for example, if you know that you're going to use your biosensor in extreme settings, then you probably want to consider something that is synthetic as opposed to a biomaterial for specific use cases.
Yeah, that makes sense. Like if it's in like extreme like pHs or extreme like temperatures or something like that, like accommodate for it.
Yeah.
And then I had like another quick one about, so like the magnetic particles that they use for the COVID detection. So they use like a biotin streptavidin, like binding complex for that. And I guess, do you have like any insight? And this might be too niche of a question for it, but like, do you have any insight on the size of those particles? And do you know like what they're made of? That like, because like there's different ones that are more or less ferromagnetic. So I'm just wondering if you know what comprises those particles, because if they're too big, sometimes they like can create a net where it actually like pulls down your I know it in the context of cells, but it can pull down your cells of interest with those beads. It like creates a net and pulls away the thing that you don't want it to pull down with it. And so I was just wondering like how big those beads are.
So most probably the beats are, I'm not familiar with this exact paper. Um, but I've seen, the most of the beats that are being used, the magnetic beats are in the order of the 10 to 30 nanometers. So these are the nanoparticles that are normally being used for this specific sensor and for this specific application. And the complex is exactly the one he told, the one he said. So the streptavidin is the one that this group used. And I would say this is actually a niche application. So probably something that I would have to come back to you, because it's a really interesting use of these particles. I would say that magnetic particles are not that used in sensor development. It's kind of like a niche, material to use. And so this is actually a quite interesting paper that I found. And I haven't exactly focused on, the actual development of the sensor. For me to highlight here from a pure device perspective was, the idea of using something that was already available, as for example, this glucometer, and turn it into something, basically repurposing to basically turn it into something that was able to do a different detection, a completely different detection, just by using, an Eppendorf tube and an already available glucometer. So yeah, really interesting approach. And let me come back to you with something, more specific about, the approach that they use in terms of like the functionalization of the surface with nanoparticles.
Yeah, that's legit. 10 to 30 nanometers is like pretty tiny. So that makes
Yeah.
sense. Yeah, thanks.
Yeah, no problem. Thanks for the questions. Okay. Well, if there's any other question, so if there's no questions, um, but if you want to go back and look at the slides again, and hey, Molly, yeah,
Oh, I was just gonna make a couple comments. Like, I really like your presentation, especially. I didn't know there was something about the coronavirus too that they were doing with just the saliva. That's interesting. Yeah, just looking at from the clinical lens. I think like on the clinical side, we'll need more studies where when we have all this data available from these biosensors and what it will mean. Like just an example of how continuous glucose monitoring has changed the care of type 1 diabetes versus, just at point checking of glucose for people with type 1 diabetes. Similarly, there are other parameters now. People have their blood pressures and heart rates available all the time. And what that means versus just a measurement once a month in a clinician office. And I believe much more focused and better care. And at the same time, I feel like it's going to be a big learning curve for the providers as well as the patients. Just thinking about newer medications, and when we try to start it on patient and, oh, you have to do this injection at home. And, that just kind of terrifies a lot of people, or there is a new pin that just clicks and it's an injection. So when these biosensors, I think it will be a lot of education on the patient's part that will be, helping. But yeah, overall, I like your presentation. Well done. Yeah.
Thank you, Wally. Let me touch on both of your comments. So the first one is basically we'll need more studies on the clinical side to see to test the efficacies of the sensors. I completely agree. So something that we're still lacking, honestly, is on the analyte side. We would need to do many more studies where we have larger basically batches of samples, and we can tell what are the specific analytes that we should target for a specific pathology. So here, the bottleneck is crazy enough to say the bottleneck is not really on the technology side, because we've already demonstrated that we are able to detect analytes through sensors up to the attomolar range that is basically 10 to the minus 27. And so the technologies there is more like, okay, now what is a panel of biomarkers that we can tell for sure if we find these markers in these specific concentration, that we can tell you have this pathology or not. And that's something that only the clinical side of it can really tell us. So a lot of studies are hopefully going that direction in the future. I would like to see some many more studies where results are compared across cohorts of people to get some meaningful data on the biomarker side. And the second comment that was also really, really important, and it's something that I'm actually keeping in mind for my personal research, is we need to develop tools that are, first of all, easy to use, because people need to use it if we wanted to use it, that do not scare people, and that are even enjoyable to have. So I'm thinking about the Oura ring, and I'm planning to get one, because I think it's really cool to have to be always in have the opportunity to see what's going on in your body, for some specific levels of markers. And so we also want people to get excited about the biosensors. We want people to be happy to continuously know what's going on in their body. And so a lot of work needs to be done in the education and in creating devices that are easy to use and people are happy to use them, because they're getting the information that they need to get, and they don't have to, we're making their life easier. And at the same time, they're more in constant, in contact with their body, more often than what they would do if they were going, to the lab every 2 to 6 months to do some specific tests. So yeah, really important comment, and something that those, the people developing the sensors always need to keep in mind, that, we have to face something. We have to create something that can be used in an easy way. Well, thank you. Thank you all very much for the questions. I just wanted to say I'll probably I'll publish I'll upload the review that I used today for most of the figures on ResearchHub. So if you have any other question that will come up later, just put a comment under the post, and I'll be happy to reply to the question. But yeah, thank you very much. That this has been really a lot of fun, and I'm looking forward actually to hear the other presentation in the series. Catch you all, guys.
Bye, Ricardo.
Yeah. Bye.